Sometimes during replication or just spontaneously, DNA will encounter errors that need to be repaired. And there are two different ways that DNA can be repaired. One of them is during replication. During the process of DNA replication, every once in a while, it's possible for the incorrect base to be placed in the new strand. And the way that that is repaired is that DNA polymerase III, which is the main DNA polymerase, it has an exonuclease attached, and this acts as a proofreader. And so when it detects an error, what it does is it just removes that sequence and replaces it with the correct bases. And so during replication, you're using an exonuclease that is attached to your DNA polymerase. And that's how repair happens during the replication process. However, there are other mutations that can occur, and there are a lot of different ways in which mutations can occur. They can be caused by water, which is kind of unusual, but every once in a while it will spontaneously happen. The reason why is that, remember, in order to join nucleotides, you do a condensation reaction that releases water. But remember that hydrolysis can also happen, and so you can use water in order to break those bonds that were formed. And so water itself can result in the loss of bases due to hydrolysis. You can also have chemical damage, which causes permanent structural changes in the DNA, as well as radiation damage, which cuts the phosphate sugar backbone of the DNA. And in order to repair this, there are a few different approaches that can be used. You can do base pair excision, where you simply remove a single base. And then you repair that using DNA polymerase and ligase, essentially. The polymerase places the new base that is correct, and the ligase then attaches that to the rest of the strand. You can also do a thing called short patch excision, and that is where you cut out a segment of the DNA that is erroneous. And you cut that out, and then you just have helicase that then unwinds the strand again, and then polymerase comes and fills in the segment. And that's another way that you can repair mutations. And these two approaches are not something that you really have to have a deep understanding of going into your MCAT, but these can often show up in passages and things like that. So be aware that if mutations do occur, there has to be some way of identifying them, not something that you're responsible for having memorized. And also the ways in which this repair can take place, either through base pair excision, where you simply excise one base pair and then replace it using DNA polymerase and a ligase, or short patch excision, where you cut out a patch of the DNA and then repair it with polymerase once again. So during replication, there is an ongoing proofreading process. That's something you should be very aware of. The exonuclease is attached to the DNA polymerase group, and so it proofreads and it corrects errors that occur during the replication process. However, if mutations show up later, and there are various sources of this, then there are a few different approaches that you can use that allows you to remove the damaged or erroneous mutants and replace those with the correct bases that need to be there.